At the heart of asexual reproduction in protists lie the mechanism of cell mitotic division. There are different variants of mitotic division in that organisms. All known types of mitosis can be found in protists. There are diplo and haplomitosis. There are different variants of location of mitotic spindle, which can be straight or curved. Moreover, the cell fission can be binary or multiple. In both cases, the cell can be divided completely, or so-called residual body can remain, which does not contribute to the volume of the progeny cells. However, the majority of the protist life cycles are characterized by the genetic recombination taking place during the sexual process. The earliest way of sexual process is the fusion of two cells morphologically identical to the feeding stages, trophozoites. In this way, some haploid chlorophyte cells fuse, producing a zygote. In fact, vegetative cells, trophont, function as gametes. Such type of a sexual process is called hologamy. The zygote nucleus is diploid, since it formed by the fusion of two haploid nuclei. After the zygote is formed, the reduction division, meiosis, occurs simultaneously with the cytokinesis, and 4 haploid cells, trophons, are produced as a result. Reduction division occurs immediately after the zygote formation, and in that case is called postzygotic. That is postgamous or postzygotic cell division. Significantly more often, specialized cells are formed to perform the sexual process, the gametes. The most simple way of gamete formation, during which morphologically identical cells are formed, was named isogamy, and the fusion of such similar gametes is isogamous copulation. Gametes are formed by the pregamous or prezygotic division. In haploid flagellates, pregamous division is represented by haplomitosis. After the formation of the zygote, meiotic division, postzygotic, is observed, during which haploid cells are formed, which become trophons. Cell division with the formation of gametes prezygotic in some species can lead to the formation of 2 types of gametes. These gametes noticeably differ morphologically, and copulation is possible only between gametes of 2 different types. This version of the sexual process is called anisogamy. Finally, maximal differentiation of the gametes is observed in so-called oogamy. Oogamic gamete copulation accordingly. During oogamy, significantly different gametes are formed as a result of prezygotic division. Immobile macrogamete, egg, with a supply of nutrients in the cytoplasm, and motile small microgametes. The function of the macrogamete is to form a diploid zygote and to provide nutrients to the cells formed after the postzygotic division. In diploid protists, the formation of gametes, prezygotic division, as in multicellular animals, occur in the form of meiosis. Haploid gametes are formed in the following way. This is the case with the actinophrida heliozoans and Coleophora representatives. The trophons are diploid in both of these cases. Haploid pronuclei, analogous to the gametes in Coleophora, are formed in the process of conjugation of individuals when a cytoplasmic breach is formed between ciliates participating in the sexual process. The reduction division of the macronuclei of each individual results in the pronuclei, one from each conjugant. The reduction division of the micronuclei of each individual results in the 4 haploid nuclei, 3 of which are resorbed, and 1 undergoes haplomitosis. Haploid pronuclei, 1 from each conjugant, migrate to the partner cytoplasm and fuse there with the second pronucleus. As a result, nuclei formed in both individuals become diploid again. We should note that during the conjugation and sexual process, there is no increase in the number of cells. Therefore, here we definitely have the sexual process, but not the sexual reproduction. It should be noted that in the case of oohamic copulation of haploid species gametes, the increase of the number of individuals also doesn't occur, because although many microgametes are formed as a result of prezygotic divisions, there is only one macrogamete. Thus, from the 2 individuals of the previous generation which have entered prezygotic transformation or division, there will be still only one zygote. All this makes one suppose that the sexual process is first of all a way of genetic recombination and possibly reparation of genetic apparatus, but not as a way of increase the number of individuals, not the way of reproduction. Even more convincing is the presence of some protists with a so-called autogamy, the fusion of gametes formed by the same parental organism. On the picture you can see the stages of a heliazoan actinophrys and its sexual process. After the diploid trophon enters sexual process, its nuclei divide mitotically and meiotically, forming only 2 gametes with haploid nuclei. The fusion of gametes and the subsequent fusion of nuclei restores the diploidity of the nucleus in the cell. It is the cell which, without the period of postzygotic division, becomes a trophon. Combination of asexual reproduction and sexual process can present in one and the same life cycle, like for example we see in the life cycle of Eimyria. For a number of protist groups, only asexual reproduction is known. For example, many amoebozoans, euglenozoa, trichomonads, and life cycles of such protists is represented by the succession of asexual reproductions. Genetic recombination is absent, and in fact all the descendants of one individual are a genetic clone, and each morphospecies is a system of such clones. Such cycles are called agamos. And finally, we need to notice that in most of the lineages, one or another form of sexual process is present, and most probably this feature is going back to the last common ancestor of the eukaryotes.